I'm Dan Kastner, as I think many of you know, and it's really a pleasure to welcome you all this afternoon to the last in the series of Wednesday afternoon lectures for this academic year before the beginning of the summer holiday season. So in any case, it's my enormous pleasure to introduce this week's Wednesday afternoon lecturer, Dr. Jules Hoffman. Dr. Hoffman is a true pioneer in elucidating the effector mechanisms of resistance to microbial organisms in Drosophila, work that has profoundly influenced our understanding of the innate immune system in higher organisms. Dr. Hoffman was born in Luxembourg and received his PhD in biology from the University of Strasbourg in France in 1969. Over his career, he's held various positions in the French National Research Agency, CNRS, the latest being that of Distinguished Class Research Director and member of the Board of Administration of the agency. From 1993 to 2005, he was the director of the CNRS Institute of Molecular and Cellular Biology in Strasbourg. The research of Jules Hoffman has continuously centered on studies of the development and the defense reactions of insects. Starting with the grasshopper, he has gone on to bigger and better things with the Drosophila, and since 1990, Dr. Hoffman and his laboratory have explored the potent antimicrobial mechanisms of Drosophila as a paradigm for the innate immune defenses. Their discoveries elucidating the role of toll receptors have had a profound impact on our understanding of the innate immune system in human health and disease. Jules Hoffman has authored or co-authored over 250 publications and edited several volumes. He's a member of the French National Academy of Sciences, of which he served as vice president in 2005 and 2006, and president in 2007 and 2008. In addition, he's a foreign associate of the U.S. National Academy of Sciences and the American Academy of Arts and Sciences. He's won numerous prizes, including the William B. Coley Award, the Robert Koch Prize, the Balzan Prize, and the 2011 Gardner International Prize in Toronto. Please join me in welcoming Dr. Jules Hoffman to the podium.
Thank you very much for those warm words. And ladies and gentlemen, I'm very honored. I was very honored when I received this invitation to give this afternoon lecture. I understand it's the last. I'll try my best to make it not the least. And what I would like to do is, so thank you. What I would like to do is show you or tell you the story which led us to understand some of the basic aspects of Drosophila immune response. And I would like to put this into a perspective, into a phylogenetic perspective, into evolutionary perspective. So a question which I'm often asked is, why did you start working on the immune response of the fly, of insects in general? Well, insects, as shown here, are frequently attacked by various microorganisms, fungi, bacteria, viruses, protozoa. I worked all my life on insects, and this cartoon here was drawn by my father, who was a professor of entomology in Luxembourg. And we started out asking what could be the defense reactions in these insects against those various microorganisms. Well, it was known, it had been known for a long time, that phagocytosis was an important aspect of a defense. We know now there's also an epithelial response, and then finally there is the systemic response. I would focus my presentation on the systemic response, which was one which has been the center of our work over the last 20 years. Also, some of our colleagues now work on the epithelial response. So, to start with, in very simple terms, let me show you when you prick a fly here at time 0 and you take off the blood after the various time intervals, you see the appearance in the cell-free hemolymph, so we're not speaking about phagocytosis here. You see the appearance of potent antimicrobial activity. So we asked 3 questions. So this is in the late 80s, early 90s. Number 1, what is the identity of the molecules which account for this inducible antimicrobial activity? From the earlier work of Hans Bohmann in Stockholm, we suspected, who had worked on butterflies and had found the cecropins, antimicrobial peptides, so we suspected these molecules to be peptides. The second question we wanted to address then is, if they are peptides, so how is the expression controlled during the process of infection? And finally, we wanted to address the question, how does the insect realize that there is an infection going on? And can it discriminate between various types of aggressors? So to make a long story short, over several years, we ended up at finding that in the fly, there are 7 distinct families of antimicrobial peptides which are induced, and we gave them various names like drosomycin, antifungal, mechnicovin, antifungal, diptericin and rosacin are antigram negatives, and atacin and cecropins are homologous to the molecules initially identified by Hans Bohrmann in the butterflies. So the take-home message here is that following an infection, or pricking, septic injury, you induce in the fat body, which is the equivalent of the mammalian liver, you induce the expression of 7 families of antimicrobial peptide genes, and these molecules are secreted into the blood where their overall concentration reaches very high concentrations in the order of 500 micromolar. I'm often asked if these molecules can have potential roles in therapy, and we may come back to that later, maybe in the discussion. So from this point on, we went to ask how are the genes controlling these antimicrobial peptides controlled during the infection. Now, at that time, we were lucky to find, and the first gene we had cloned was the diptericin gene, we were lucky to find kappa B response elements. Similar results were obtained also for the cecropin genes by our Swedish colleagues. Now, NF-kappa B had been identified a few years earlier by David Baltimore and his colleagues, and so when we mutated the sites very precisely and looked at the expression of reporter genes, we could see that mutating the kappa B response element would abolish the inducibility. So they were really mandatory for the induction of the antimicrobial peptides, our readouts for the defense here. So very shortly, the fly has 3 NF-kappa B family members which go by the name of dorsal, I'll come back to dorsal in a second, and diff, which stands for dorsal-related immunity factor. Both molecules have this REL homology domain which we are familiar with in a mammalian system, and they are retained in the cytoplasm by binding to the inhibitor cactus, which is the equivalent of I-kappa B. Now there is one third molecule which was identified by Dan Haltmark, which he named relish, which has the ankyrin inhibitory domains on its C-terminus, and which requires not dissociation from the inhibitory domain in contrast to dorsal and diff, but proteolytic cleavage, which will then free the REL homology domain and associating transactivating sequences to go into the nucleus and to control expression of antimicrobial peptide genes in this case. Now, the problem of NF-kappa B here was that at the initial stage only dorsal was known, and dorsal was considered as a maternally expressed gene. And let me shortly show you, the dorsal had been identified by Nüsslein Vollhardt in Germany and her colleagues in the embryonic system. So they had shown through mutating genes in the mother that maternally expressed genes controlled the, among other things, the dorsoventral axis in the embryo of Drosophila, and they showed that this was mediated by the dorsal gene, which was inhibited by the action of the cactus gene. At that time, neither dorsal nor cactus had been cloned. I've presented here dorsal as an NF-kappa B family member, which became apparent a few years later, and cactus we now know is, we have known for quite a long time, is an I-kappa B equivalent. Now, the system dissociates upon receiving a signal from a transmembrane receptor, which Nüsslein Vollhardt referred to as toll even before it was cloned, just referring to the phenotype of the mutated embryo. And toll, via various intermediates, activates phosphorylation of cactus. Cactus becomes degraded in the proteasome, and dorsal can translocate into the nucleus. Now, how does toll become activated in this system? Toll becomes activated by a cleaved growth factor, which they refer to Spätzle, Again, all these terms were created by Nusslein Vollhardt and her colleagues and refer to the phenotypes of the mutated embryos. And so Spätzle itself becomes cleaved as the end product of a proteolytic cascade. I'm not insisting on the names here, but so a trigger in the follicular cells, which is not fully understood yet, will activate the proteolytic cascade, and cleaved Spätzle, which we know now is a cytokine in our system close to neurotrophins, will then activate the toll receptor, and we go to the transcription of embryonic genes by the NF-kappa family member dorsal. So far for this introduction. When we got the results which I've mentioned earlier, when we cloned the genes, we thought that it might be that this embryonic cascade, which was considered as maternally expressed genes, a cascade of maternally expressed genes, might also play a role in the immune response in controlling the expression of the antimicrobial peptides. What was against this idea was that, as I mentioned, these genes were maternally expressed genes. So we first went on to show that they were also, dipterosin as illustrated here, was also expressed later in development. And importantly, we could see that an immune challenge was upregulating the expression of the dipterosin gene. Now, in this slide, what we have done is look at the expression of the induction of dipterosin in the wild-type context by a challenge with bacteria and fungi. And as you see in the wild type, we get a very nice induction. RP49 stands for as the loading control. And I must say, to our utter disappointment, was one of the greatest disappointments I remember in my scientific career. In the toll deficient background, there was no effect. Fortunately for us, although we didn't realize the importance at that time immediately, we had a line in our stocks, a mutated line, which later on we refer to as IMD, in which there was no induction of the dipterosin gene by this immune challenge. Now, when we found the drosomycin antifungal peptide 2 years later, and when we tested drosomycin, we had the converse phenotype. That is to say, this time we saw that drosomycin was not inducible anymore in the toll deficient background, but dipterosin, but was, fully inducible in the IMD background, which then indicated to us that we had probably two signaling pathways controlling the expression of the antimicrobial gene families, one which would be a partial reuse of the toll pathway of embryonic development, and another one which we proposed to call IMD pathway, although at that time we had nothing in our hands except for mutant fly, IMD standing for immune deficiency. Now, importantly for us, at that time we could show that if we were trying to infect flies, and if we infected flies with E. coli, as illustrated here, we had, you will see the red line shows survival in wild-type flies. They are not affected, wild-type flies, not really affected by this E. coli in the conditions which we're using here. These are not the aggressive E. coli strains of northern Germany. But when we compare the survival to in the IMD mutant background, you see that it's dramatically affected. In other words, really, the IMD pathway plays a role in protection against gram-negative bacteria. Remember that it induces the expression of dipterosin, which is directed against gram-negative bacteria. Importantly, again, such flies would survive normally the infection of, as wild type, the infection by fungi or by gram-positive bacteria. And in the second slide here in this series, I show that in a toll deficient background, we're looking at here Aspergillus infection. The toll mutant flies are dramatically affected as compared to wild type. And this yielded then this picture, which has gained some celebrity, which was in our cell paper, Bruno Lemaitre, Emmanuel Nicolas, Lydia Michaud, Jean-Marc Reichart, in which we had a fly which is overgrown here by hyphae. This is a toll deficient fly which was infected by Aspergillus. Now, let me just pause a second and say, so we came from the Drosophila background, and we had a collaboration at that time, a Human Frontiers grant, which I was coordinating with Charlie Janeway, Al Nazekowicz, Fotis Kafatos. And so let me just point out why this was important at that precise time. So it had been described, was known that antigen-presenting cells were activating in this case T cells. So this is an antigen-presenting cell, which is, and this is a naive T cell. So what was known is that when microbial ligands, however we call those, interact with this antigen-presenting cell, they activate NF-kappa B via an unknown hypothetical receptor. And then through NF-kappa B, there's an upregulation of MHC expression, of co-stimulatory molecules, of cytokine production, and so on. So this ends up, this aspect of innate immunity ends up by stimulating adaptive immunity. So this was a paradigm of adaptation, of activation of adaptive immunity by innate immunity, which was available at that time and had been proposed particularly by Charlie Janaway, as you may know. And now, let me show you the results put into perspective of that period. So on one side of the slide, we see here CD14, which was known at that time to be able to bind LPS, lipopolysaccharide, one of the prototypical inducers of innate and adaptive immunity. So, but CD14 is GPI anchored. And we're looking for something, everyone was looking for something which activates NF-kappa B. So this would not fit, although it was an interesting indication. On the other hand, it was known that the interleukin-1 receptor signals to NF-kappa B via a domain here, which is the T domain, but it has extracytoplasmic IG-like domains. Did not correspond to what was really being looked at. And so now, in here we show toll. So toll seems to fit into the pattern of the leucine repeats on which in CD14 bind LPS, and it has, interestingly, intracytoplasmic domain, the T domain, which is really very close to the one which in the interleukin-1 receptor activates NF-kappa B. So what this then suggested is that there was, at least in one metazoan system, there was a chimera of the extracytoplasmic domain of leucine repeats capable of binding LPS, for instance, and intracytoplasmic domain capable of signaling to NF-kappa B, and that this chimera was capable of helping the fly defend itself against a microbial aggression. Now, from there on, as you may know, several studies in a very short period. So our study was published in '96. 1 year later, then Charlie Janaway and Ruslan Medjitov, with whom we collaborated at that time, and when found a mammalian homologue of, a human homologue, I should say, of the Drosophila toll, and they could show by transfecting the intracytoplasmic domain and the transmembrane domain in human cells hooked to an extracytoplasmic CD4 domain, that they could activate NF-kappa B and induce an adaptive immune response. And a year later, Bruce Beutler, by cloning the LPS gene in mice which were non-responsive to endotoxin, he could show that it was in fact the extracytoplasmic domain of this receptor was a domain which reacted to LPS and then conveyed a message to NF-kappa B via the intracytoplasmic domain. And in parallel, or a little bit later then, Shizuo Akira in Japan, and he did a series of very important studies in which he clarified the ligands of the mammalian equivalents of the fly toll, which are referred to as TLRs, and as shown here, they all bind to microbial ligands, be it LPS, lipopeptides, flagellin, and there are other of these receptors which are not cytoplasmic, but which are not on the cytoplasmic membrane, but which are in the endosome and which respond to various nucleotide sequences. I have no time to go into detail of that, but that has been very well established, in particular by Shizuo Akira. Now, as a result, we have via adapter proteins, MID88, TRIF, TRAP, TRAM, activation of NF-kappa B, and in the mammalian system, but not in the fly system, IRFs, and then production of antimicrobial peptides and activation of adaptive immune responses. But let me come back to the fly now. And so we initially thought, of course, that toll might react to microbial ligands. It took us quite some time to show that this was not the case, but it was the cytokine Spätzle which played a role in the embryonic development, which was binding to toll, activating toll, and itself was cleaved by a proteolytic cascade, which was induced during fungal infection and gram-positive infection. At that time, it was clear also that the fly had 9 tolls, not only 1 toll, and so the idea was close, of course, that we were in a situation which would parallel that found in mammals, that is to say that various of these toll receptors would bind to various sort of microbial ligands and help distinguish between various infections. This was not shown to be the case, and up to now there is no demonstration that any of these other tolls plays a role, at least, to state it carefully, in the systemic immune response as I have described it in the first slides. Now, so this left us with the question now, how are these molecules, how are these microbial ligands recognized if it is not the toll receptors? So it was Julien Royer in the laboratory who in 2000, by doing unbiased mutagenesis in flies infected with various microbes, generated and isolated a mutant fly, which he referred to as Semmelweis, in which gram-positive bacteria were not inducers anymore of response, and the flies were dying from simple Streptococcus infection. When he went on to clone the gene, we found that it corresponded, that it was a member of a family of genes which had been earlier, a few years earlier, identified again in butterflies in Japan, and which were named after their capability of recognizing Peptidoglycan. I'll try to be short on this. There are 13 members in the fly. The important thing is that they all have here a site, so a groove, which is an amidase groove, and this is conserved in many of these molecules. Actually, I should point out that those molecules which are this protein family, which is referred to as peptidoglycan recognition proteins, that those molecules are ancient, evolutionary ancient amidases, already present in bacteriophage. And some of these, so I said they're circulating molecules and transmembrane molecules. Some of these have lost their amidase activity, and they have transformed from a catalytic function into a recognition function. And I'll illustrate this in the next slide. And so this would be peptidoglycan, with which we're all familiar. This is a chimera. It's an artificial molecule. And in the position 3, in gram-positive bacteria, we have a lysine amino acid here. In gram-negative, we have diaminopimelic acid here. Now, what the amidase does, of course, is cleave between the sugar moieties here and the stem peptides. And this renders peptidoglycan non-immunogenic. This is a way for the fly to decrease the immunogenicity of peptidoglycan. Now, when this is a zinc coordinate, which is coordinated here, a zinc ion. Now, during evolution, in some of the members of this family, this coordination has been lost through mutation in the surrounding amino acids. And then we have here an example. This would be the Semmelweis family. And it could be shown that this specifically recognizes the lysine form of peptidoglycan, that is to say, that which predominates in gram-positive bacteria. However, another form, resulting also from this mutation and loss of zinc ion, will recognize specifically the diaminopimelic acid, that is to say, the one which is predominant in gram-negative bacteria. So in all this has shown, let me just point out to the names of the people. There are several laboratories which were involved in this. We have not directly been involved in the biochemical part which has solved this problem. So let me now go to a slide which will summarize what we have learned about the recognition of microbes in the system here. So gram-positive bacteria interact with one of the forms of this peptidoglycan recognition protein. There's an association with another molecule which I will not go into. And then there's a series of serine proteases, a whole cascade, which will go and which will activate the so-called Spätzle processing enzyme, which is shown here. And that will then activate, cleave Spätzle and activate Toll. So this is the case of a gram-positive infection. A fungal infection interact with another protein, which is called glucan binding protein, which is illustrated here. It is not identical to the PGRPs, but it is well characterized now. It's of the same size, and it has glucan binding domains. And this will then activate this, again, this same cascade and lead to the activation, the cleavage of Spätzle and activation of Toll receptor. And finally, and this is an interesting point which has been worked out, this has been essentially worked out by Bruno Lemaitre and also by Jean-Marc Reichert. And here, this Dominique Ferrandon and Jean-Marc Reichert have shown that microbial proteases can activate this system via a dedicated circulating zymogen, and let me just, which they name Persephone. Let me just say a little word on this. I think this is really important in as far as we know that most of the microorganisms do secrete proteases. This also is the case for protozoan infectious agents. And those proteases, in this case, this was shown here in the case of Bovaria bassiana. And in this precise case, where there were mutants available on the fungal side, mutants on the fly side, our colleagues were able to show really that fungal protease interacts with the circulating zymogen of Drosophila, cleaves it, and feeds into this cascade, leading to Spätzle cleavage and to activation of the Toll pathway. So this would be in general terms of discussions which have been around in the field for nearly 20 years. This would be a case of recognition of microbial ligands, and here we would have a case of recognition of endogenous abnormal molecules, the cleaved Persephone. And to finish with this aspect, gram-negative bacteria interact with another member of the protein, peptidoglycan recognition proteins, LC, which is a transmembrane receptor and activates directly this system, the IMD cascade, to which I'll come in a second. So, having established now, first of all, some of the essential effectors of the immune response of the fly, that is the antimicrobial peptides, having come close to a point where we understand how those microbes can activate the cascades which lead to the expression of the antimicrobial peptides, let us turn to these cascades now. That is to say, let us look at how NF-kappa B is activated by Toll in Drosophila. So we have seen the microbial sensors for fungi and gram-positive bacteria, GMBPs or PGRP-SA. They lead to the cleavage of Spätzle. Spätzle interacts with Toll, and then Toll will interact with MID88 or MID88, which has been conserved also in mammals. So we have two tiered domains here, and this is the DEF domain, and then association with the kinase PELA. And also it is established, it seems to be that this kinase domain is not the one which is phosphorylating cactus. Cactus becomes phosphorylated. We should say that we do not know yet which kinase really does this effect. And from after phosphorylation, cactus becomes degraded, and either in the larvae dorsal or in the adult diff, translocate to the nucleus, where they control the expression of the drosomycin gene, which was our readout, but of hundreds of genes. There are hundreds of genes which are induced in this process, and this has been studied by various microarray analysis in various laboratories, and also most of, I should say 50% of the genes which are induced are a function which we cannot clearly relate yet to the immune phenotypes. So this would be for the activation of dorsal or diff by the Toll pathway. It's also known, I have no time to go into this, also there are a certain number of informations are available how the system is turned down at a given moment. One of the ways is, of course, to increase among the genes which are regulated is the cactus genes. It's the cactus gene, which will then, of course, contribute to stop the system. Then there are serpins upstream of Spätzle, which also are induced in response, and they will lower the inducibility of, or the cleavage of Spätzle. Now, let me turn to the second pathway, NF-kappa activation by the IMD pathway. As I mentioned, but maybe too rapidly in the beginning, there is a large NF-kappa family member in the flies, which goes by the name of relish, which is equivalent to mammalian P105, which requires proteolytic cleavage. Now, as I mentioned, gram-negative bacteria via their diaminopimelic acid form of peptidoglycan will interact with the PGRP-LC transmembrane receptor, and then a series of events occurs which is relatively complex. I try to be as simple as possible. In fact, there's first, there is the FAT homolog of mammals, which is present here, and DRED, which is homologous to mammalian caspase 8. Now, there occurs a process here in which DRED will cleave IMD here, and then through IAP2, which is an ubiquitin ligase, we will have activation, we will have ubiquitination of IMD as illustrated here. This was nicely worked out by Neil Silverman here in the United States in association with our laboratory. And then we have TAC1, this MAP3 kinase, which binds to this, which probably, that's what the idea which is in the community now, will bind to this chain, as will the fly equivalent of NEMO, IKK gamma, which is shown here. So there is an IKK complex in the fly here, an equivalent IKK beta and IKK gamma. There's no equivalent IKK alpha. So the idea which is in the community is that we have as a result of the association with peptidoglycan and its receptor, a series of events occur which probably all occur upstream here. And this chain which is extended here may just be in the proximity of the cytoplasmic membrane up here. And through ubiquitination, we'll associate various partners here which play a role like IKK gamma, TAC1, the kinase, and of course, after ubiquitination of IMD, which is shown here. IMD has a DEF domain, and interestingly, this DEF domain is closest to the one which we find in mammalian RIP for TNF receptor interacting protein. Now, what then happens is that once IKK beta has been phosphorylated, this complex will phosphorylate relish, which is illustrated here with the inhibiting anchoring domains, and this is the dimerization part, nuclear translocation part, which will contribute to the expression of the responsive genes, whereas this part remains in the cytoplasm. So as a result, we have the expression very rapidly of dipterosin, and again hundreds of genes whose functions remain relatively uncertain in the context of the immune response of the fly. I should also add that downstream of this MAP3 kinase TAC1, as in the mammalian system, we have activation of the DRN kinase pathway, which does not participate in the induction of dipterosin, but in that of cytoskeletal proteins and pro-apoptotic signaling. So I thought it could be helpful if I summarized what I've said about these two pathways, which is something which is in our eyes essential, in the following cartoon. Let me put the two pathways one next to the other. So this is the toll pathway in the fly in the context of the immune response. So upstream, and this is important to notice, we have proteolytic cascades. This is in contrast to the situation found in mammals, where you would have direct interaction of the microbial ligands with the toll receptor. So microbial ligands can induce this. Of course, also developmental cues, as illustrated here, because that's the way Nissla and Follard discovered the role of the toll receptor. And then, as I've pointed out, proteases or virulence factors, if we wish, like illustrated in the case of Persephone, can also activate the system. So it's a platform which integrates various systems, various cues according to the developmental situation or physiological situation. Then via diff, retained via cactus, so you could say here P65 and I kappa B, this system becomes dissociated, and we have activation of gene expression. On the other hand, the IMD pathway In this case, we have a transmembrane receptor. We do not have an upstream amplification cascades. The microbial ligands directly interact with an IMD pathway receptor, which we know in the case of DAP peptidoglycan is PGRPLC, and then relish through very complex situation, cascade, relish becomes cleaved, and this part will go into the nucleus and will induce immune response genes. Now, a point which I would like to make here is that there is good evidence now that this pathway, it's probably not a developmental pathway as far as we can tell, in contrast to the toll pathway, which is probably initially a developmental regulator. This pathway is really much more a response pathway to infection, but also to stress signals and to to series of let me say unfolded protein problem, to neurodegenerative signals, for instance. We know that, for instance, in fly models of Parkinson's disease, this pathway is induced. We know also that in aging flies it is induced. We know that in DNase 2 deficient flies, and this was work of Nagata laboratory in Osaka, the pathway is induced. So that's the reason why I've put here endogenous ligands. And Polly Metzinger, who's in the audience, would say that these are damage signals and they induce this pathway with this, which is a possibility. So I wanted to show the slides to make the distinction clear. Now the question, do those pathways, do they crosstalk? There is no clear answer to that. There are, of course, some reasons to believe. I mean, if you really push one pathway, you will just by as I said, by squelching the proteins which are involved on one side or the other, you will have an effect, but it is not clear, it's not established yet, whether there is a crosstalk, and I'm not convinced that this molecules, the NF-kappa family members here, can heterodimerize, as has been occasionally proposed. So I would now like to take this information to have a look at the evolution in the last part of my presentation, the evolution of this system. So, of course, when we started doing this work, we wanted to see, at that time, I really must say very frankly, that when I tried to convince the director of the Life Sciences Department in our research agency, he was very reluctant to fund this work and saying that we don't know if the fly has any immune response, and we don't know if this is relevant. And then again he said to me, which was harsh, and if someone should take this up, it should be a medical doctor and not you as a zoologist. And so I would like to put now the information which we got over the years, to put it in perspective with what we have learned in other groups. So this is the toll receptor interaction with, can you see this? I hope you can see it. In association, well, interacting here with the Spätzle cytokine, and then I've just put a few of the members. I'm not going to take the whole cascade. It will blur the information. So we have MYD88. We have a kinase here. In this case, it's a pelakinase, but as I mentioned, we're not sure. It's the one which is phosphorylating cactus, and here's cactus or I-kappa B associated with N-kappa B, and then after dissociation, will activate gene transcription. So this is the case of toll. Now, let's put TLR4 here. There's one first difference. In TLR4, it is not a cytokine, but it's LPS, which will interact. Then we have MYD88, we have ERAC. To some extent, we can put this equivalent to PAL kinase. We do not have TEC1 in this series here. We have activation gene kinase, an IKK complex, and then I-kappa B, NF-kappa B, as pointed out. Now, let us turn to IMD. Now, in the case of IMD, we have, as in the case of TLR4, we have induction by a microbial ligand. Here is peptidoglycan, here is LPS. If we had selected another TLR, we could also have put peptidoglycan there. So we have here IMD, we have TEC1 feeding into gene kinase pathway, the IKK complex, and here what I've explained to you, activation or cleavage and activation of relish. And then finally, let us put here TNF. Now, you will see that TNF, which will react to a cytokine, as was the case for toll, has downstream RIP, which is equivalent to mammalian, to Drosophila IMD. We have TAC1 feeding into gene kinase. We have the IKK complex, and again, the rest is equivalent with some slight modifications. So this is the four pathways, this is a selection of the two pathways in the fly and the major pathways in the mammals. And you see how conceptually this system is equivalent, which of course no one anticipated in the early 90s. Now this then begs the question, when did this appear? So we don't suppose that the fly was the ancestor of mammals. So when did this system appear in nature? And in the next slide, I'll show something which I could not have shown a few years ago. This is work which was done in Boston by Tom Gilmore and John Finnerty and their colleagues. And they were looking at the genome, the many genomes which have been sequenced in the last few years, and many more are coming out. And they were looking at the sea anemone Nematostella. And they found, and in this slide, this was a part which dates 2 or 3 years back, they found an equivalent in the genome sequence of TLR, with mid-tier domain, an equivalent mid-88, the equivalent IKK, of TAC1, of NF-kappa-B, and I-kappa-B. This came as an enormous surprise. It was not totally convincing until recently. The same group and other groups in Germany this time were experimentally showing in cell systems that in Nematostella the toll could really direct the system, as I've shown in the fly. There was via TAC1, IKK was dependent on the system, and NF-kappa and kappa. So they established experimentally that the sequence, as we have seen them in the fly, that the sequence also is valid in Nematostella. Now then, we come to the question, when did all this appear? In this slide, I've tried to summarize the evolution as one might see it. So multicellularity has appeared 1 billion of years ago, probably. You can say 1.2. And we have the protostomes illustrated here and the deuterostomes illustrated here. So sponges are among the most earliest life forms which appeared in the Precambrian. And then we have a separation here of the sea anemones from the rest of the protostomes. And the insect protostome, the time levels are given here. Of course, around 800 million years, there was not much going on in evolution on the Earth. There was the time of a snowball. And in the Precambrian, the explosion really occurred in Precambrian, particularly in the Cambrian. And so we have insects, worms, and mollusks appearing, echinoderms here, and then the chordates, hemichordates, and the chordates a little bit later, 450 million years ago. So let's just ask the question, as we have now a lot of evidence available, particularly experimental evidence now in sea anemones, and I believe in sponges this will come out. Let's ask the following question. What are relevant witnesses of these cascades? So when did they appear? So when did toll appear? When did NF-kappa-B appear? When did TAC1 appear? And when did the effector molecules, antimicrobial peptides, appear? And in just one click, I'll show you the answer. They appeared very early on. In the sponges, we have antimicrobial peptides, NF-kappa-B, TAC1, and toll. And they're present all along the evolutionary range. So, in other words, this indicates to us that in contrast to what the community believed, innate immunity is a very ancient process, not only in terms of fighting in a mystical way, invading microorganisms, but also at the molecular and cellular level. Now, what then has changed? Why aren't we all relying on innate immunity? So let me center 1 second on the deuterostomes. So echinoderms, the chordates here. And so what has appeared to the surprise of everyone is that in echinoderms you have 222 toll-like receptors. You have 203 node-like receptors. I have not mentioned node-like receptors because they are not present in the flies. So they are intracellular receptor molecules involved in a variety of defenses. Particularly, they play a role in Crohn's disease. That's why they have become so famous, and also in adjuvancy. And then there are 218, these are scavenger receptors rich in cysteines. There are only 6 of these in the fly. So in echinoderms, you have a fantastic explosion of those receptors. When we look at Amphioxus, which has been recently sequenced, we see still a very large number, 48 TLRs, 92 NLRs, 270 of the scavenger receptors. So when this appeared, a first explanation which was provided by in the community was that maybe those the echinoderms and Amphioxus live they live certainly in a sea medium, and there may be so many unexpected pathogens coming up, and that's why they need so many of those. I should say, experimentally, none of these molecules has been documented to play a role. So they are just, this is genome sequence. Now, this proposal that the large number of pathogens would explain the situation with a large number is certainly not valid, as Siona, which is the eucordate, which lives in the same situation, same conditions, Amphioxus and Strongylos and Trophus, has only 3 TLRs, has 0 NLRs like the fly, and 22 scavenger receptor rich in cysteines. So this is puzzling. This is puzzling, and it's probably too early to speculate on what has occurred here, and instead I want to go with you rapidly through the last point I want to make here. So what has really changed between all those groups and us in terms of true vertebrates? So what has changed is, as I've put here, what is certainly absent from all the other groups is the sometime here after the first whole genome duplication, there was a bifurcation of the lymphocyte lineage from the myeloid lineage. This has been analyzed and discussed in much detail in that community, in the specialized community. And development of long-lived lymphocytes with the potential for self-renewal and selective clonal expansion. And this has really been the major change. It's a change of cell biology, first of all, at the basis of the chordates, and then we have the separation, of the hagfish, the agnathans, lampreys, and the other groups which have full mouth skeleton, the gnathostom. And both groups have, but independently, in a different way, developed those massive recombination of antigen receptors. In the case of the gnathostomes, that is to say, the fish, true fish, and all the tetrapods, it is through B and T lymphocytes, through the famous VDJ recombination, which is also, it's not only a complex system, but it's a very variable system. It's not a really homogeneous, in all the groups, there are many variations which allow to do this VDJ recombination. And on the other side, we have many people have involved in these studies, an enormous host of immunologists, and on the other side, you have essentially the work of Max Cooper, and Max Cooper here in the US has shown that there's another type of recombination which he refers to as the VLR, variable lymphocyte receptor recombination, and also 2 types of lymphocyte-like cells. We speak here of lymphocyte-like cells, A cells and B cells, which are equivalent to the B and T cells. So this is really what makes the distinction between us, I'm including fish with us here, between the vertebrates and the invertebrates. And the vertebrates have kept the innate immune system still as a very important tool of defense. But of course it does not show memory. And then my very last slide will give you the philosophy of I think it's wrong to say that the fly, we can look at the fly to understand how the adaptive immune system has evolved. The fly does not have an adaptive immune system. The point I want to make here is that at a given moment, all those groups, they were all separated after, nearly all after the Cambrian explosion. And if we give 100 million years more, they were all separated. So you see, all of these groups have evolved according to different constraints, but starting from the same pool of basic molecules. And should I say molecular modules. And so you see, for instance, Nematostella has kept nearly everything. So Nematostella is as close as you can get to a human in terms of immunity, of molecules involved in immunity. It does not have recombination. That has uniquely appeared, as I've mentioned in the preceding slide. C. elegans has lost nearly everything. There's no toll receptor involved in immunity. There's no NF-kappa B. and still they survive. But not as well, of course, as we do. Horseshoe crab has NF-kappa B, has toll receptors, but does not use them in the same way as Drosophila. And Drosophila has lost totally the NOT receptors, whereas there are many of these, about 70 in the metastellas, and as we have just seen, about 200 here in the echinoderms and in Amphioxus. So each group has, from a common background of molecular models, has selected its own ways following the constraints and the options it had in its evolution. And with that take-home message, I would like to stop here and say that it has been a fantastic period because, of course, of this. None of us suspected that we would come to this sort of understanding at a given moment. Of course, it's fragmentary, and much of it is still hypothetical, but I think we have a fairly better understanding that we had 20 years ago. And all this work was done by many of my excellent colleagues in the laboratory and also by other groups, and I just would like to acknowledge quickly. So I started this work with my wife, and she was also working on grasshoppers like I was at that time initially. And Charles Huttrue as a chemist and Jean-Marc Reichert, now the head of the laboratory. There Jean-Marc Reichert is a geneticist, as is Dominique Ferrandon here. Bruno Lemaitre was for several years with us, and he was particularly involved in the toll work. Julien Royer in the PGRP work. Jean-Luc Imler is doing the studies now on antiviral defenses, in which I participate, I should say. The AMD pathway is also involved in the antiviral defenses, but that would be another seminar. Elena Devashina has worked in the laboratory on plasmodium and plasmodium reactions. And then this would be credit given to a series of colleagues whose work I've integrated into the journal slide, particularly Neil Silverman and Tom Magnatis in the United States, Catherine Anderson and Tony Ip and David Schneider, and a long list of, and here Sarah Cherry, Raul Andino, that would be antiviral defenses. Linda Stewart would be phagocytosis, which I have not mentioned. In Europe, it was essentially the group in Stockholm, which has initially worked on the butterflies, and then other people whose name I've mentioned already, and in Asia, Kurata in Sendai, and Lee and Kim in Korea. So thank you very much for your attention.
Well, thank you very much, Jules, for that fantastic seminar. We have a reception that will be in the library afterwards, but perhaps if there are some questions from the group before we adjourn to the reception. I see one already. Go ahead.
So my question was, when you showed the IMD pathway and you were showing the ubiquitination, I was wondering if you ever looked at aberrant ubiquitinization, and maybe with age, if it's not enough or too many of the ubiquitin molecules, that might actually interfere with the pathway.
I'm sorry, I'm not certain I understand your question. You're asking?
With the IMD,
yes,
you showed the ubiquitin molecules.
Yes, yes.
So if you have too few or too many, do you actually get aberrant activation of NF-kappa B, which with age could actually make sense?
Yes, that may be so. That may be so. You're right, with age, the insect spontaneously activates the IMD pathway. And I've been quick on that, but I realize I've already been too long. So the system is much more complex. If you give me just 20 seconds and not more, we have done a proteomic analysis now of molecules binding to the 11 canonical IMD pathway members. And we have isolated 320 partner proteins which interact with this, of which about 150 give a phenotype upon RNAi. 50% of those are conserved in the mammalian TNF pathway. So you see, this is just the beginning of this, and I didn't want to overload you with, but you're perfectly right. This has to be investigated.
So, Jules, here. So as you've seen for the Drosophila, with the number of toes, you have over 10 toes, then The other toes, there's no known function for them. So
Oh,
perhaps it
I'm
would
sorry, I should say they are all involved in embryonic development.
Okay.
All of them.
for
Yeah.
immunity.
Yeah. But not in
So-
systemic immune response, yes.
So perhaps it's a little misleading or confusing to look at the receptor, a number of receptors in different species, and maybe it would be more insightful to look at the effector mechanism, or in terms of look at the antimicrobial peptides that might be encoded in the genome, because those are direct effectors in the immune response, versus the receptors, the number of receptors, some of them may be redundant, may have no non-function.
Yeah, you're perfectly right. Let me just answer, say the following thing. Number one, antimicrobial peptides are present in all the groups, and now recently, which is funny, recently a group in Germany has isolated in humans drosomycin-like peptide. We produce drosomycin-like peptides. So that's true, I mean, and that has been done. So antimicrobial, that's right. And then regarding the toll-like receptors, giving the numbers of 222, and so you're perfectly right. And as I mentioned, I pointed out that it has not been investigated whether they really do play a role. They're just there. And
Just too many of them. And
yeah, of course. But it has been investigated. What has been done is in the sponge. What is being done in the sponges now is to see is it a developmental regulator or an immune regulator, and we're still waiting for those results.
So you find this commonality going back to the multicellular animals, but that's as far back as you mentioned. So what happens if you go back even further to single cellular animals or plants, and even back further to prokaryotes?
Yeah, it's an important question. And number one, let me point out that there are antimicrobial peptides produced by bacteria. And this is well known, so bacteria do fight in our gut, they fight each other with antimicrobial peptides. It is not clear, as far as I can tell from the literature, if something in protozoans, the production of antimicrobial peptides, and it has been looked at, but it has not. Would you know, Alan, data on that? No, to my knowledge, it has not been found, but not in the way we looked at them then.
Well, so if it were me, as opposed to you, I wouldn't be looking, even though that's what Sam suggested, for the antimicrobial peptides. I would be looking for the systems that respond to them, because I think those systems also respond to other forms of damage. Right?
Yes.
So rather than looking at war, how
Yes.
about looking at healing?
Yes, you're right, you're right. It's something which will have to be done, and we'll keep it in mind. And but it has not been done. Yeah, it has not been done so far, that I would know.
Maybe one more question.
It's brilliant.
So Jules, what's so striking is in vertebrates, the TLRs, we often refer to as microbial sensors.
Yes.
The striking thing is, of course, in Drosophila, the toll is not a microbial sensor. It's a cytokine sensor, if you like.
Yeah.
And we have, and Drosophila has used an entirely different system to detect the microbial threat and to transmit that to toll. So you showed us there were 3 sets of recognition elements. Do you have a feeling whether that's the total, or that the recognition elements that will lead to the cleavage of Spätzle
Yes.
is larger?
So Spätzle is a member of the Neutrophin family in the fly. There are 6 members like that. They're all activated during development, including Spätzle, the immune Spätzle. So I would say that the pathways which I've illustrated here are probably the end story in terms of systemic immune response, because you can combine, as you can also in the mammalian, so you can combine various mutations from various pathways, and then it's it. Say if you combine a mutation in the toll pathway, mutation in the AMD pathway in a given fly, it will not resist in any way. So that's why I believe this is the end of that story. Now, I should point out that in the mammalian system, there's much more to it than there's also the PGRPs, for instance, which I've mentioned here, play a role in our immune defenses. You may not be aware of that, but we use them also, particularly as spectrostatins. And did that answer your question?
Well, I guess my point would be the three elements, the three sensors that you would describe.
Yes.
Are they so central?
Oh, I
the-
see.
That
I see.
that the
No, yeah, I see.
microbial
No.
agent cannot
Yeah.
mutate to escape them.
I see, yes.
Yeah.
I see. No, there would be much more open-minded. I mean, upstream of Spätzle, what will activate the Spätzle processing enzyme, it may be that there are many other molecules which have escaped that. But those, that's not as easily to point out in terms of genetic experiments. But it can be done, but it's more, you have to use sensitized screens.
Sorry, I just said, okay. You said in Drosophila that microbial proteases can activate the toll-like receptor signaling pathway. In humans, do you know if matrix metalloproteases
Yeah.
can also activate the TLR
Absolutely.
system?
Yeah, absolutely. You're perfectly right, and we were looking into that direction. But okay, we cannot do everything and not this, but it would be very interesting if someone took this up in the mammalian system. Absolutely. Absolutely.
All right, well, I think that with that, we'll call it to a close. Thank you very much again, Dr. Hoffman, and we'll have a reception in the library.